Hey guys, we're going to be taking a look at the next topic, which is titled Metabolism and Growth. So this is going to come from parts of chapter 7 and 8. Now I'm going to have you do some textbook reading over just 7, and 8 I'm going to cover in the lecture, but we'll actually cover parts of 7 in the lecture also. So I'll kind of cover both in the lecture, and then you'll do reading just on chapter 7. So getting started, we're looking at the idea that the activities in a cell constitute what we refer to as metabolism, and we'll get to that definition here in a second. But the way that anything in general happens within a cell takes place with the action of a molecule called protein. So we're all familiar with protein. We think of it as a source of nutrients, a type of nutrient. But really on a cellular level, what proteins are doing in a large extent is conducting metabolic reactions, and they are more or less the source of our metabolism. So we can think of them as the quote-unquote workers of the cell. And worker just means something that's going to get something done, initiate a reaction. Over here to the right real quick, this is a diagram that shows in a general way kind of how metabolism works. So food, nutrients come in, and they go through a series of reactions that break down molecules in what we'll talk about here in a moment called catabolic pathways. So this is a breakdown. We generate some building blocks, and we generate some energy, and that also releases heat. Most importantly, the building blocks and the energy are used to then reconstruct new molecules, and then these become able to give us more nutrients and more energy to sustain the cell. So this is kind of the sum total of all metabolic activities in a nutshell. So ultimately, each step of the way, we have proteins as the workers conducting these, and that's what gets things done. Now, to understand how these actually work, we want to start by understanding that proteins themselves are made up of individual units called amino acids. There are 20 different amino acids. These are, generally speaking, the 20 amino acids used by virtually all organisms. And every organism, therefore, uses these to build proteins. Proteins come in a lot of different shapes and sizes. There are millions of different types already recorded. And it's estimated that there are possibly hundreds of millions of types out there in the world. So there's more or less no limit to them. But what's unique about each is the way the amino acids are constructed and the way they're put together. So what's important about that is to understand the way a protein works. You have to understand how it's built. And so it begins with these amino acids. Now, amino acids are shown over here on the right, and they are a group of molecules that have similar but unique properties. So if we look at an individual amino acid, what we see is that they have two types of bonds on either side that are consistent in all 20 amino acids. Let me pause it real quick and pull up a better view of that just a second. Okay, so this is from website nutrientsreview.com. So we'll just pull this here. So what they're showing is an individual amino acid with a breakdown of their chemical bonds. So we've got carbon, we've got hydrogen, we've got nitrogen, and we've got oxygens. So it's a basic molecule. Now, the key things about this molecule is that with all proteins, we have an amino group here and a carboxyl group here, sometimes called a carboxylic acid group. So these two are found on all 20 amino acids, in addition to a hydrogen right here in this location. That's not key to our discussion here. So the two key groups are the amino and the carboxyl. The other group is called a side chain, and this is where each amino acid is actually different. So if we go back and take a look at this, we can see that each of these amino acids have a different R group or an R side chain here. So this is what makes each one behave differently. Okay, so the key thing about these individual, or excuse me, the groups on each individual amino acid, the carboxyl and the amino, what's key about them is that they allow any two amino acids to form a bond together. And what ends up happening is that the carboxyl group of one will automatically link to the amino group of another, and vice versa. So this amino group could link to this carboxyl, and this carboxyl could link to this amino. So any of the 20 amino acids are universal in that they can form a bond with each other like this. So what that allows in nature is for all of these amino acids to join in big long chains like this, and then the way that they're arranged starts to change how the actual protein behaves. In other words, the difference in each protein is the exact way these 20 amino acids interact to form a chain. So there are like as I mentioned, millions of different proteins, each arranged in a different way. So just go back here real quick then, and just to point out that the properties of a protein then are the arrangement of the amino acids. More specifically, it's the exact types, which of the 20, how many, and then the sequence of those 20. Now, the quantity varies. The smallest proteins are around 100 amino acids. The largest can be pushing close to 40,000, in the upper 30,000s. So really, really large differences in between the size. Most are several thousand typically. So we're talking about several thousand generally linked together in a chain. So they get really large typically, and it can get extremely large. Now, so here I've shown just a small chain to represent how they would link together. There's a bond called a peptide bond that links those. And then this becomes what's called a polypeptide, meaning many peptide bonds. So this is a zoomed in view here to show that this would be a much bigger chain. And then what this is showing is how what happens at the next part of the discussion here is that the chain actually starts to bend and twist into a three-dimensional shape. And I use the analogy here, for those who remember the pipe cleaner exercises that you did in grade school, where you took the pipe cleaner and you would bend it and twist it into three-dimensional shapes. You know, let me just pull up an image of that real quick, just a second. Okay, so some of these are actually pretty good, but these all start as a little pipe cleaner, and you just a straight little wire with bristles on it. I'm sure, hopefully you know what I'm talking about. But these can then bend and twist into a three-dimensional shape. And ultimately you can form some pretty elaborate shapes apparently with those. Nothing like I used to do. Mine were very simple. But anyway, the point is it's an analogy to show how a protein kind of folds in a similar way. So this starts out as a chain and bends. Now, what actually makes it fold is the way each of these amino acids actually interact with each other. So for example, you have polar amino acids. They will be attracted to each other, generally speaking. Nonpolar will be repelled by the polar. So in certain areas, the chain will actually bend towards itself, and that can make a certain type of shape. In other places, it will actually repel away from itself, and that will twist it in a different direction. So the chain actually is through attractions and repulsions based on the amino acids will actually bend and twist into its shape. And ultimately, this forms a three-dimensional shape. And here we're just kind of filling in some of the space to show that ultimately this takes on a three-dimensional form that starts to have a function. So one of the things we'll talk about next is that or actually it's mentioned right here, excuse me, the shape of the protein determines its function. So I mentioned earlier they're kind of like workers. Another way to think of them in this case is think of them as tools. And it kind of works in both ways depending on the context. But here you can almost think of it as a tool where the shape of a tool determines its function. So think of a hammer that hammers nails, and it has a very specific shape to hammer those nails. And then you've got a screwdriver that screws, and it's got a shape to do that function. Well, every protein is similar in that its shape will determine exactly how it functions. And that's where each one of the different, millions of different proteins will be a little different in that shape, and then also ultimately that function. Now, this is my amazing drawing here, in the artistic ability maybe just a little lacking. So this is from the web, and it shows a little better three-dimensional idea of what these proteins might actually look like. So these are computer models. These are not the real thing, but they're computer models showing how these proteins would twist and form different shapes here. So there's different names for each one, and they all have some kind of specific function within the cell. So that's a bit about the proteins there. So ultimately, they run our metabolism. So, and back to the metabolism part of it then, metabolism is defined as all the chemical reactions and physical workings of the cell. It's really a broad term to just describe what's happening in a cell. Most of the things that are occurring are in direct relation to the metabolism, although not all, most. So, there's two general types, and these are very broad categories, but one is considered anabolism, and you could refer to these as anabolic reactions. Now I think of this, and this is how I remember it, think of anabolic steroids, because anabolic steroids build you up, they build muscle. Anabolic reactions are also building. They're considered biosynthesis, and they create structures. So you can see over here an anabolic reaction where smaller molecules combined with an input of energy allow these to be joined together to form a larger molecule. So it's building things up, biosynthesis. The other type is called catabolism, and you refer to these as catabolic reactions. It was right there the whole time. So catabolic reactions will start with the larger molecules and then break them down into smaller molecules and then release energy at the same time. So they're basically opposites of each other. And if you just go back real quick to the first slide, you see here that we start with the catabolic pathway, which breaks things down, and then that releases the building blocks and the energy, which then are reused to drive the anabolic pathways. And you might ask, why would they break things down just to rebuild them? Well, think of the food as like a salvage yard. You've got a lot of stuff here that you can use, but it's not in a functional form. So like a salvage yard, you have to break down the parts of like an old car, take out the good stuff, and then rebuild it into something that you can actually use. Your body's doing a similar thing. The food you have has all the carbon and all the hydrogen, all the elements that you need to drive your cell, to meet the cell needs, but they don't work in that exact form. So you have to break them down, create the building blocks, and then rebuild them into the exact form that your cells need. So that's, in a nutshell, what metabolism is all about, breaking down your nutrients and recreating the exact things that the cells need. So proteins do this, but getting a little bit more specific here, it's actually a type of protein called an enzyme. So let me just clarify that enzymes rather, are a type of protein. There's lots of different proteins. Proteins are a fairly broad class of molecule, so not all proteins are enzymes. And it's actually true that not all enzymes are proteins, but to be fair, most enzymes are proteins. And I don't mean for this to be confusing. I'm sorry, I'm rambling a little bit here. There's an exception. You do have RNA enzymes, but those are actually a recent discovery. If you're interested in modern science, this is actually a relatively new thing. We never got taught this when I was taking this about 10 years ago. So RNA enzymes is a new class. It's somewhat new. I'm not really all that knowledgeable about them, but a traditional enzyme is a protein, and these are the more specific type of molecule that functions in metabolism. So an enzyme then is really defined as a substance that is a biological or biochemical catalyst. So here's the full definition: a substance produced by a living organism that acts as a catalyst to bring about a specific biochemical reaction. So it's a biological catalyst is basically what it is. So then the next thing you need to know is, well, what's a catalyst? Well, a catalyst is defined as a substance that increases the rate of a chemical reaction without itself undergoing any permanent change. So in other words, it's something that initiates a reaction and then can do that reaction over and over and over again. So it's not consumed, meaning it can be used over and over again. And this goes back to the tool analogy. Think about an enzyme or a catalyst being a hammer, and a hammer can hammer a nail and then hammer another nail and do it over and over and over again. So the hammer is not consumed in the process of hammering the nail, therefore it's free to be used over and over again. And so just to contrast that, some things can increase the rate of a reaction, but they are consumed in it. And an example of that would be heat. Heat can be used to speed things up. It's actually how we cook food. When we cook food, we're essentially catalyzing not catalyzing, excuse me, but we're speeding up some of the chemical reactions that ultimately make it safe to eat and change the nature of the molecules. But we're doing that with something that we don't get back. So heat is a consumable type of reaction, whereas an enzyme is non-consumable. Now you have other types of non-biological catalysts. An example would be a catalytic converter. If I can spell out there we go. Catalytic converter is something that you would have on a car. And this is actually an emissions control device that breaks down some of the pollutants that come out of a car. So one of the ways that modern society has cleaned up air pollution. Now, in this case, it's a type of metal, I believe. I'm not all that knowledgeable about it. I probably shouldn't even say. I'm not sure exactly how it works. But it's a non-biological catalyst, and it is a similar thing where it breaks down molecules in a way that can be used over and over and over again. So it's a different type of catalyst. So that'd be a non-biological catalyst. So enzymes are biological, and they're unique in that sense that it's an organic type molecule that can do this type of reaction. So the cell ultimately produces lots of enzymes, and then they start to carry out the reactions, and then the cell then ultimately reaps the benefits by collecting some of the energy and some of the benefits that come from those enzymes. So here's the first review. Go ahead and pause this, and we'll move on to the next part here in just a second. Okay, so in the next part, we're going to look at how the enzyme is actually creating a reaction, or what it's actually doing. So one of the next things we want to understand is that when it comes to an enzyme, they're always going to have another molecule that they interact with called a substrate. So a substrate is a type of molecule that interacts with an enzyme. And depending on what type of enzyme it is, there's one of two things that will generally happen. So many enzymes are considered catabolic enzymes, and in that sense they break down a substrate. So remember that a catabolic reaction is a breakdown reaction. So a catabolic enzyme is therefore a breakdown enzyme. So it takes a single substrate and it breaks it down into two smaller substrates. So in this figure here, what we're looking at is when the reaction is going this direction, so from this to this, we're seeing a catabolic reaction. So one substrate enters and it breaks it into two smaller pieces. On the other sense, then we have what's called an anabolic enzyme, and it in that sense does a synthesis reaction, or it combines two smaller substrates into one. So in this figure here, you're looking at it going from right to left, where we start with two substrate and they join together to create one larger substrate. So many enzymes function in one of the other directions here. So there are exceptions. The book talks about different types of enzymes. If you want to read about that, I'm not going to require it, but chapter 8 goes into greater detail on more elaborate types. But in a large number of cases, many enzymes function in this kind of way. So break things down by taking a molecule and breaking it apart, or build a molecule up by taking two smaller ones and joining them together. So that's the idea behind a catabolic and an anabolic enzyme. Now, the way an enzyme and a substrate interact goes back to what we said in previous discussions, where we talked about lock and key specificity. Now, lock and key specificity applies in this case where the enzyme acts like the lock and the substrate acts like the key. And the enzyme has a shape that facilitates the binding of the substrate to this particular location. And this location is called the active site. So the active site is a specific region on the enzyme that has a shape that allows the substrate to interact with it. Now, some things happen in relation to interaction, but in general, that's the shape fits into the enzyme's active site here. Sometimes the enzyme will move and conform to the substrate, which makes it a little different than a true lock and key analogy. But in general, there's a kind of a shape fit here that goes together. So we have a groove-like pocket, we call it the active site, and it has a lock-like shape that allows the substrate to fit in with key-like specificity. So very similar to what we've talked about before. So just to connect this back to what we talked about earlier, then it's ultimately then the sequence of amino acids, and the amino acids form that shape, and this shape then determines what the active site looks like, and then that determines what substrate it interacts with. Okay, now if we're talking about a catabolic enzyme, it's going to interact with one larger substrate and then ultimately break it apart. If we're talking about an anabolic enzyme, it's going to interact with two smaller substrates and it's going to join them together. So the active sites would look a little bit different, but ultimately the same general rule applies, that the shape is going to allow these substrates to fit together here. So that's where the structure goes along with the function, excuse me. So the function in this case, just to clarify, the function will be to interact with the substrate. Now if we just want to connect that one more time here, just to make sure stay grounded with the overall big picture, some of these enzymes then are going to be breaking down food into building blocks. That's what a catabolic enzyme would do. Take a larger substance, break it down into smaller substances. An anabolic enzyme would then grab a couple of these smaller substances and join them together to make larger molecules. So that's all, again, we're generalizing here, but that's to connect back to the overall picture here. This is a quick video that gives you a quick visual on everything I just talked about. So nothing dramatically different, just a an animated perspective on that. Let me turn my volume down real quick here, just a second. Actually, not sure how this is going to work. Let's try it here.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme called the
Sorry, let me try that one more time.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme called the active site has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
What he meant by that was that if you do this in reverse, you'll see one substrate come in and break apart into two smaller substrates. So here's an anabolic reaction is what they were showing. Two substrates come in, they join, they combine. Anabolic. If we're looking at a catabolic reaction, it actually starts like this, except you would change the word product to reactant, or substrate rather. And so the substrate would come in, be acted upon, and it would break it apart. So that'd be a catabolic reaction. So either way, and that's where I kind of drew that figure earlier showing the two arrows going in opposite directions, because they are essentially opposite processes. The next video here is going to show how this applies to an example in the human body. It's going to talk about the enzyme sucrase, which breaks down table sugar, or what's known as sucrose. So sucrose is the substrate, sucrase is the enzyme, and they sound very similar because they were named that way. The enzyme was named after the substrate, which, by the way, is actually fairly common. I don't get into enzyme naming here, but the name of the enzyme often coincides with the substrate that it interacts with. So, okay, so here's the video on sucrose and sucrase.
An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.
Okay, so it's showing you a more specific example, putting some names and context to it. But once again, we see here, in this case, a more elaborate active site. And you can see here how the active site takes on the shape of the substrate, so it fits in there with this lock and key like specificity. Now, what this was showing is one of the ways that an enzyme often works, especially a catabolic enzyme, and really where the lock and key analogy fails a little bit, is that really when an enzyme binds to the substrate, it usually moves and contorts with the substrate, and it acts in a way that actually stresses and actually bends the substrate in many cases. So in this case, what's happening is that a water molecule, when the enzyme bends the substrate, the water molecule will break those bonds. And so the only thing I like about this video is it shows this water molecule floating in here like a magic carpet or something. But what's really happening is that water is all over the cell. So this enzyme is immersed in water because most cells are mostly water. So there's always water all around it. So as soon as the enzyme puts a stress on that bond, there's a water molecule right there. And the way the chemistry works is that the water molecule actually facilitates the breaking of that bond. And so it actually takes the enzyme plus the water molecule for the full reaction to take place. So this is an example of how that would work in relation to table sugar. And so just to clarify why this would happen, sucrose is a relatively large molecule, and your body can't absorb that directly. So the way your body actually absorbs this chemical is by first breaking it down into two smaller components, one of them being glucose, the other one being fructose, and then they can be absorbed. So once they're in their smaller components, the body can absorb them. But in the larger sucrose form, it's not absorbable. So this is called a disaccharide. These are called monosaccharides. Okay, so anyway, move on. That's an example there of how that would work. Now, another factor that goes along with many enzymes are what are called cofactors. So a cofactor is something that facilitates an enzyme to function properly. So we're adding another layer here. So we start with the basics and then we kind of build upon that. So the truth is that most enzymes don't function totally independent. They don't function independently with just the substrate, rather. They need an additional molecule to go along with it, and there's a whole bunch of them, so they're generally referred to as cofactors, which is a kind of a broad term to describe a molecule that interacts with the enzyme. Now, the way that they generally work is they facilitate the binding at the active site. Another way to say it is they kind of complete the active site. So in this example here, it's showing the enzyme here and the substrate here, and they want to fit together with that lock and key specificity, but they don't quite match up. And this figure doesn't show that all that well here to start with, but that enzyme doesn't quite match with that substrate. Now, when we put the cofactor in this exact spot right here, that allows the substrate to then bind more appropriately. So in a way, it really kind of completes the shape of the active site and therefore allows the substrate to bind. And that's really kind of generalizing what is a much more complex and a much more variable process. So lots of different ways that it actually works. But in general, that's more or less what's happening. So a cofactor then is really, so the key thing here, by the way, is a cofactor then becomes necessary. And in a lot of cases, these have to come from the environment. And there's an assumed evolutionary connection here where these enzymes evolved in a coevolutionary type scenario with these cofactors involved. So that they evolved in a way that they only work with what was around in the environment, which were these cofactors. So cofactors, to connect this to what we're already familiar with, cofactors, generally speaking, are what vitamins give us. So many vitamins, what they actually do for our body is they act as cofactors. And what they do then is they allow our enzymes to function properly. And that's why we need vitamins. We need our enzymes to function properly. We need our metabolism to be working correctly. And in order for all that to take place, we need the enzymes to ultimately be functioning properly. And so it just turns out that in nature, that usually only works well when you have the cofactor to go along with it. So when you look at the back of a vitamin bottle, you see a lot of different examples of cofactors. And so I didn't add all that information here, but that's something to connect to your diet. So cofactors are important. Now, you don't want to overdo it with vitamins. You actually get many of them from your food. Now, I'm not going to get off on vitamins, but generally speaking, if you eat good natural food, you'll get plenty of vitamins. Only when you eat lots of processed food would you probably want to start taking multivitamins. Most nutritionalists would only recommend that if your diet doesn't, if you don't have a good diet, more or less. So, okay, so another layer in reference to enzymes is that many of them have an additional binding site that adds another layer of functionality, and this is what's called a regulatory site. Now, you can think of the regulatory site a lot like an on-off switch. That's how I like to think of it. It's basically like a little button on the enzyme that when you push it, the active site will either turn on or turn off. And there's different ways it can go either direction. So in other words, it can start by being on and then it can be turned off, or it can start by being turned off and then turned on. So once again, you could draw, I didn't draw the arrows here, but you could watch this go either direction. So it could start off, and so here's a situation where the regulatory molecule hasn't bound to the regulatory site yet, so it's off. So it can't bind the substrate. Regulatory molecule binds, that opens the substrate, opens the active site, turns on the active site. Now the substrate can bind. Regulatory molecule releases, active site goes back off. So we could then reverse that, where it starts off and then turns back on. So anyway, the regulatory site, then, to clarify, is dictated by the regulatory molecule. So it has a similar lock and key specificity, just like the substrate and the active site, but in this case it's a different spot on the enzyme and it's a different molecule with a different shape. So that's why I drew this figure here showing a different type of interaction. So it's not the same as the substrate. It's a different type of molecule, and it has a different type of shape to act with a different spot on the enzyme. But really just think of it as an on-off switch, and the molecule acts as the button pusher. So the regulatory site is the button, and the regulatory molecule is what pushes that button. And depending on the enzyme, it'll either turn it on or it'll turn it off. And that's the way a regulatory site works. Now what that does, to clarify, is it gives the cell some control over an enzyme. So they serve to control the actions of enzymes. Now, I'm not going to quiz you over all this, but just to give you a little bit extra information here, there's really two ways a cell can regulate an enzyme. So regulation of enzymes really occurs in two general ways. And one is, I would call, this is not the technical term, by the way, but I would call it pre-production. And what that means is that it's control at the genetic level. The genetic level, I mean control of the DNA. Oops. So in this case, you're basically controlling whether the DNA creates the enzyme. So whether the enzyme is even made or not made, pre-production. So if you need the enzyme, you make it, and then it is then used. Now, many enzymes are actually just created without a genetic regulation. So the only way to control them once they've been synthesized is post-production, and that's where you get regulatory molecules. So in other words, when you want to control what's happening in a cell, you either control it at the genetic level, and what that means is whether you make the enzyme or don't make the enzyme. The enzyme is either made or it's not made. Once the enzyme is made and it's already been put out there, the only way to control it at that point is through regulatory molecules, and they bind or block the active site. Or they act as the on-off switch for the active site. So if the regulatory molecule binds, it turns off the enzyme, and it can no longer function. So it gives the cell a form of control on the enzyme. Okay, so here we are at part 2. Go ahead and pause this, and we'll continue on here. Okay, so next we're going to talk about how enzymes actually function in a more dynamic way. So we've really been kind of focusing on individual enzymes and individual reactions. The truth is that these enzymes are actually going to work in what we call a metabolic pathway. So they actually work in a series of reactions where one enzyme begins the reaction that is then continued by the next. It's a lot like an assembly line. You can think of a metabolic pathway a lot like you think of an assembly line, where we go back to the worker analogy, and each enzyme is like a worker. And like the worker of an assembly line, each one plays a small part in the overall process. Now, like any assembly line or any process that occurs in a series, there's generally one step that is considered the pacemaker. So they set the rate of the progression, and they can also control whether that pathway begins or comes to a halt. So whether it's initiated or shut down. So here's a quick figure showing different types of pathways. Many of them occur in a linear form, although it's also very common to have a branched form. In a linear, this is the true assembly line analogy. You have a reaction from A to B, and then that reaction proceeds from B to C, etc. Each time you'd have an enzyme and a substrate. So, for example, substrate A would be acted upon by enzyme A that would be shown by the arrow here representing the enzyme reaction. And that would create substrate B, which would then be reacted upon by a different enzyme. So each step would involve an enzyme and a substrate, and then a different enzyme and a substrate. But the key is that the product of the first substrate, or the product of the first reaction becomes the substrate for the next reaction. So if we just go back real quickly here, if we were showing it with this type of example here, this substrate, if we're going from this direction here, for example, this substrate would be broken down into these two smaller substrates. They become two separate products, and then either of those two products could become the reactant in the next reaction, and then that can continue to break those down. So really the truth is, when we go back to the very first slide and we show the overall series of reactions in the cell, the truth is we're summarizing this in an overall scheme, but each step would involve a series of pathways. So for example, in order to break down the food into tiny building blocks, you don't do that with one enzyme. You do it with many enzymes, and each one takes a small piece of the larger molecule and it's broken down in a stepwise fashion. So imagine a car being disassembled for its components, as I analogized this earlier. So the car wouldn't just be magically taken apart in 100 pieces in one step. You'd have to take the car apart one step at a time. And that's essentially what you see with a pathway in the way enzymes work, is they work in little pieces and each one contributes to the overall whole. So this figure doesn't show all that, so I want to come back and clarify. This would, in order to be more accurate, need to show lots and lots and lots of little steps. Now the problem with that is they get really complicated, they have all these funny names, and they get really specific, and I really personally want to avoid most of that, since this isn't really a class that's meant to emphasize advanced biochemistry. I want to just kind of talk about the more general aspects without really getting into the details of all these different pathways. So if that's what you want, if you like that kind of stuff, if you're into that, you want to know more about that, that's really what biochemistry is all about. Biochemistry really is the detailed study of all these different types of pathways and different examples, and you get into the study of all the enzymes and the substrates, and you can imagine how much fun that is. But we're going to skip over that. So now just to talk about this for one more second, generally speaking, you'll have a pathway that's linear, and in this case, A ultimately is substrate A is converted to a totally different substance through this series of reactions. A branch pathway is a scenario where one substrate can actually be acted upon by different enzymes. And so this is an exception where you can actually have a single molecule be acted upon in different ways. And so in this sense, you have kind of alternate routes that a substrate can go down. So if it can go down one pathway and be converted to one series of reactions, or it can be going on a different pathway and can be converted through a different series of reactions. So that's kind of an example of a divergent. A convergent is similar in that two different pathways can create the same molecule. So we can arrive at a single molecule from two different pathways. And this is part of the redundancy of cells, to generate the same things through multiple ways, and that gives the cell more diversity in how to get the important molecules. So these are usually critical to the cell survival. A cyclic pathway is a little harder to describe, but you can think of it as a wheel that needs momentum in order to rotate. And that momentum is given by an incoming product. So there's usually a series of enzymes, and what they do is they take an incoming substrate or an incoming reactant, and they convert it through a series of reactions and ultimately generate byproducts that the cell can use. So think of this as like an outcome of the reaction, something valuable that's produced. But then ultimately, the starting substrate is regenerated and the pathway begins again. That may not make a lot of sense. I've never been good at explaining these. They're probably one of the more complicated. But in order, you have to really look at the specifics to understand these. But basically, they work in a way that you have an incoming product, and that allows the series to cycle, and ultimately you can generate the starting product again at the end, and it starts over. So the reactions don't actually line up in a circle in the cell. The reactions here are considered cyclic because the end product is also used back in the beginning once again. So you generate the beginning product at the end, in the pathway cycles in that sense. So that's why it's considered cyclic. Anyway, I'm not going to really emphasize too much detail on that. So if it doesn't make a lot of sense, I apologize. But those are, like I said, really need the more specific detail to understand those. So if you want that, by the way, I would recommend looking up what's called the Krebs cycle. And the Krebs cycle is an example of a cyclic pathway. So that's an example that would give you some more information on a type of cyclic pathway. I'm sure all of you will go look that up right now. Okay, here's another video showing how a biochemical pathway works.
Organisms contain many different kinds of enzymes that catalyze a variety of different reactions. Many of these reactions, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, a substrate is converted into a product by the first enzyme in the pathway, and the product of the first reaction then becomes the substrate for the next reaction. The product of the first reaction then becomes the substrate for the second enzyme. The sequence of reactions continues until the final product is made. When a biochemical pathway is functioning, the initial substrate is continually converted to the final product through the series of steps in the pathway.
Okay, so there's a animation showing a pathway in motion there. Now, going back to what we mentioned earlier about regulatory molecules, in that same kind of context, we said that an enzyme can be turned on and off. And what really happens, to connecting this to the pathways, is that what really happens is the pathway will actually regulate itself through the production of regulatory molecules and what are referred to as competitive inhibitors. So the way this works, let's go back to this figure here, to this part of this animation here. We can see here's a miniature pathway, 4 enzymes, each catalyzing a different substrate. What ultimately will happen within a cell is that these pathways just run autonomously. There's no brain in the cell. And what can happen is that the cell can produce too much of a certain product. And you can almost think of this from like an economic standpoint, using a human analogy. Imagine a tire factory that can't sell all the tires it produces. So it takes money to build the tires and create them, and yet they can't sell all of them. So, for whatever reason. So it doesn't make sense if you're a tire company to keep making tires that you can't sell. Your warehouse is already stocked. You're investing money in something that isn't selling. And as a result, you would have to stop production. But you'd have to do that with a thought process. Cells don't have any thought. There's no brains. So they've actually evolved to become self-regulating. So they have a similar capacity to turn themselves off, but they do it automatically. And it's kind of cool how it works. So in a lot of cases, what happens is that the end product, the final product of the cell, will actually act as a regulatory molecule in a way that it can turn off the first enzyme. Usually it's one of the first, if not the first enzyme in the pathway. It's usually one of the earlier enzymes. So the the final product, actually acts as a regulatory molecule. So it's not only something that the cell produces that it needs, but it also serves as a dual function to regulate the activity of this first enzyme. Now, generally speaking, that only happens if there's too much of a final product. So if this product is being used by the cell, then it won't ever build up into a high quantity. And that's kind of where I use, that's why I use the tire analogy. If you're selling tires, then you want to keep making them, and there's no buildup, and there's no problem, and your warehouse isn't full. So you keep selling them, and you keep making them. But when the economy turns bad or whatever would stop people from buying them, at that point, they start to build up. And it's the buildup that ultimately halts production. And that same thing happens here, just again, autonomously. So the final product will actually build up in the cell, and eventually what will happen is that high concentration will cause it to act in a regulatory way. It's going to be kind of hard to draw. I'm going to skip that here. I don't know if that makes sense, but think about it this way. The cell has only so much room, and as the product builds up, it starts to saturate the cell. As it saturates, it becomes closer and closer in contact to the first enzyme in the reaction. And ultimately what happens is that if it gets too high a concentration, it gets too close to the first enzyme, and at that point it begins to act as a regulatory molecule by bonding. So in other words, it won't bind as a regulatory molecule if there's not very much of it and if it's being used. But if it starts to build up in the cell, then it starts to eventually act as a regulatory molecule and stop the first enzyme. So ultimately what that does then is it shuts down that first enzyme in the pathway, again, first or one of the first, and then ultimately what that does is it stops the production of the product. Because remember that this product is being created through this series of steps. So if you shut down the first step, you never make the second and the third and then the final. So you ultimately stop the production. So it's kind of a cool way that cells regulate their own pathways, and it's a way that they can use less energy. And this is actually an evolutionary prediction. It's part of the evolutionary theory, because what it basically entails is a conservation of energy, and cells that conserve energy the most efficiently are more likely to survive. So these cells have under that theory evolved these pathways as a way to conserve energy. So it's something that we see in virtually all living organisms. It's a very fundamental part of life. And by the way, this is what we generally refer to as negative feedback. Or you could even say negative feedback inhibition. So the feedback down-regulates the process. There are examples of positive feedback where the product actually amplifies the reaction. Those are less common, however, and most of the time they're considered negative feedback inhibition. So going back to this slide here, there's generally two ways that happens. The first is what I mentioned with the regulatory molecule. And this, for whatever reason, they like to come up with these fancy names, and so they call this non-competitive inhibition. So when a regulatory molecule acts in a way that turns off the enzyme and therefore regulates a pathway, they refer to that as non-competitive inhibition. And that's because the other type of way it happens is called competitive, and in this situation you have a molecule that's similar to the substrate. It's able to actually bind to the active site, but it's not reacted upon, and therefore all it does is actually block the active site. So just think of it as a blocker, and they call it competitive because it competes with the active site. They got kind of cute in the naming here. But really all it's talking about is how the regulatory molecule regulates the enzyme, turns it on and off, and then you have another kind of a different example of how that works where the cell produces a competitor molecule. It's really, in most cases, it's the same example where the pathway creates, so this is like the final product, and the pathway then creates a molecule that acts to turn off one of the first enzymes. So it's just a different way the cell kind of does the same thing. So non-competitive regulatory, competitive competitor molecule. And also as an interesting connection to something that you're already familiar with, a lot of toxins and poisons are actually harmful to cells because they act as competitive inhibitors. Many poisons actually block the active site of an enzyme and can actually cause the enzyme to stop functioning. So there's a certain capacity of toxins to actually bind to an enzyme and block it, and therefore cause disease in the body because your body doesn't function properly. So I'm kind of generalizing there, but that's the way some, like neurotoxins are an example. A neurotoxin is often toxic because it binds to a neuron receptor and blocks the communication of neurotransmitters. So many neurotoxins act as a competitive inhibitor. So some of them are really strong in that they bind and they never release, and so they ultimately kind of kill the nerve cell. Others are less toxic in that they can bind but then ultimately release, and therefore not necessarily cause the same kind of permanent damage. But toxins can have that same capacity, and that's why certain things are toxic and other things aren't. And that's not the way all toxins work, to be clear, but sometimes they do. Neurotoxins is an example there. Okay, the next section here is going to talk about how enzymes can be affected through environmental factors. So at this point, we've kind of established, and just to bring it to the front of your mind here, that enzymes are pretty important, and our cells need these. Metabolism is all the things happening in a cell, and metabolism is run by all these different enzymes, so it's all critical. So what happens then is that the environment can play a role in affecting the way enzymes function. And really, when we start to look at the specifics of how exactly the environment affects a living thing, in a lot of cases it actually comes down to affecting the activity of enzymes. So for example, when we say we heat something up to the point that it dies, a thermal death temperature, really what that means is that we've heated it up to the point that most of its enzymes no longer function anymore. And generally speaking, we refer to this as denaturation. So denaturation is a term or denaturation, if you want to potato, denaturation. It's basically a term that describes when an enzyme loses its shape and therefore loses its function. Now, and so this figure here shows how the normal shape can be lost, and this occurs through, again, heat is a common example. Cold actually has a different way of affecting an enzyme. Acids and bases are another way that they're denatured. I'll use cold in a different context here in a second, but generally these can destroy the shape of an enzyme and make it no longer function. So when we talk about how the enzyme affects something, there's a lot of cases where it actually affects the enzymes and then therefore indirectly affects the rest of the organism. So, and just to clarify, denaturation is a term that can also apply to nucleic acids, DNA and RNA. So you can also denature DNA and RNA in addition to proteins and enzymes. So, okay, so the environment has a big role. One of the biggest, as I mentioned, is temperature. Now, there's a couple ways that the temperature affects enzymes. One way is it affects the rate of activity. Now, to fully understand this, we have to get into some of the physical properties and some of the bioenergetics, and so we're not going to do that. But to kind of summarize what's relatively complicated topic, enzymes need a certain amount of heat in order to function properly. So if it's too cold, they don't function properly, and you can think of them as being too sluggish. They don't move fast enough. They don't bind substrate very fast. And it's like a person who is extremely tired and barely awake. They're just not, they're there, but they're not functioning. So at a cold temperature, they just don't have much activity. And that's why typically when we cool things down, a lot of times they don't actually die. Now if you get to a freezing temperature and we form ice crystals, ice crystal formation can kill many things. But really the truth is, outside of ice crystal formation, cold temperatures really don't denature. What they really do is just slow things down. Now, if you slow things down too much and the metabolism of a cell stops for too long, then many things will die if the metabolism slows down to a really slow point. But the truth is, there's actually a lot of organisms that can actually freeze themselves and then come back to life once they rethaw. That actually happens in fish and in a lot of marine, not maybe marine organisms, but freshwater pond organisms. So cold affects things very differently, and that's kind of the point here. I'll move on. I don't mean to keep going on about this, but cold temperatures slow things down, become sluggish, and the rate of activity drops. Heat has a very different effect. Now you want a certain amount of heat because what that does is it speeds the enzymes up, and so they function better at a certain temperature. And so this figure here is showing that as temperature goes up, so going on the horizontal axis, we have an increase in temperature, less temperature, more temperature. So as temperature goes up, we see that rate of enzyme activity goes up as well. And there's generally a point for every enzyme that it's got an optimal temperature. And every enzyme is a little different. But most of them, it's somewhere between 60 and 100 degrees Fahrenheit, which just so happens to be the exact temperature that most organisms thrive in, and that's not a coincidence. So our enzymes ultimately are what are most affected by the temperature, and therefore we are affected by the temperature because it affects our enzymes. So really we're getting down to the fact that temperature affects our cell activity. So at this optimal temperature, the enzymes are moving around at the right speed, and that allows them to function properly. Now, if we get too high, then what happens is that we get denaturation. So the temperature, the high heat, will actually destroy the delicate bonds of the protein. And if we go all the way back to where we talked about amino acids here earlier, each one of these little peptide bonds here, and each of the bonds that allow the more complex three-dimensional shapes to form, they are somewhat delicate. And what heat does is it starts to shake things and cause them to vibrate. So heat really is vibrational energy. So as things get hotter, the molecules are actually vibrating faster and faster. So what's ultimately going to happen if you get too hot is you get too much vibration, and those bonds will actually break. And that causes the three-dimensional shape of the enzyme to start to fall apart. And generally, the more you heat it up, the more denaturation you get. Sometimes you can heat it up a little bit and it'll lose its shape, and then if you cool it down, the shape will come back and it'll start working again. But usually there's a point at which you heat it up too much and it can never reform the shape again properly. So every enzyme is different, but generally speaking, too much heat and they denature, and if it goes too far, you get permanent denaturation. Now, the truth is every enzyme is different. As I just said, we actually have some enzymes that function better at high temperatures. And so just to point out an exception, you have what are called heat-stable enzymes. And these enzymes allow some organisms to function at higher temperatures. So one of the things we're going to talk about next is temperature adaptation and how some organisms actually prefer a higher temperature. An example here is a microbe called Thermus aquaticus. It actually grows in the hot pools and the hot pools, the hot vents around Yellowstone National Park. Let me pull that up here. It doesn't sound right. The hot springs, hot pools. Okay, I guess hot pools wasn't way off. These types of things here. I don't know if you've ever been to Yellowstone, but they occur in areas where there's lots of magma below the surface, so they heat up water near the surface, and you can go here and look at these, but they contain unique microbes that actually flourish in really hot temperatures, so you can see how hot this water is. Most life doesn't prefer that. Most things wouldn't live in there, but there are some bacteria that actually thrive in that higher temperature. So that's an example of an exception, and they actually have heat-stable enzymes that allow them to do that. So it's one of the things that starts to set life apart, is the way their enzymes work, and you get unique types that you only find in certain organisms. So here's part 3. Go ahead and pause this, take a look, and we'll go on to part 4 here. Okay, so we got 2 more parts, and these last 2 parts are a little bit shorter. So this next part here just connects to chapter 7 and how different enzymes kind of control different behaviors in the environment. And really what I mean by that is that different organisms thrive in different temperatures based on the types of enzymes they have. So for example, you have some organisms that actually prefer colder temperatures. Examples would be like fish that live near the Antarctic. Not the most common types of organisms, but anything that lives in a cold temperature. Not anything, but many organisms in cold temperatures are considered psychrophiles. So they're organisms who grow optimally at low temperatures. Just skip here and go to the other extreme. Thermophiles would be like what I just showed with the hot vents. They are organisms that actually grow at a high temperature. So these are kind of both of the extremes to a certain extent. Mesophile, meso is in reference to middle or moderate, and a mesophile is something that would grow at more of an intermediate, more of a normal temperature. So most organisms would be more of a mesophile. It really, what that means is that their optimum range is within these temperatures here. Now, the truth is, all organisms have a broader temperature range in which they have a minimum in which they can't really survive past any lower than that point, and then the same on a higher point. So usually the range consists of a minimum and maximum, and then the optimum is a range within there that they grow at their best. So they can survive within the minimum and the maximum, but thrive and grow as they're kind of intended to within the optimum range. So minimum maximum shows you survival, and this shows you really where they're going to grow and actually live according to their biology. Okay, so another major factor then, so that was a bit about temperature there. Another major factor is pH, and pH is a measure of acids and bases. So pH is something that's, again, a more complicated topic, but really an acid is a substance that gives off these little things called hydrogen ion. And hydrogen ion, in a nutshell, is toxic in high concentrations. So an acid basically puts off a substance that if you have too much of it becomes toxic. A base is similar. It gives off a different type of substance, and in a similar sense, if you get too much of it, it too becomes toxic. So certain chemicals that have extreme pHs have different levels of toxicity based on those pHs. Now, not all of these are necessarily bad, but and pH is kind of a complicated thing, so I'm probably not explaining that very well. But it's basically a measure of a certain type of substance and how much of it you have. Now, if you get too much, it starts to affect the way enzymes behave. It starts to affect the way your metabolism behaves. And so really, it's a type of substance that controls the behavior and the functions of cells. So pH, acids and bases, really have an effect on the way a cell functions. Okay, so what they ultimately do is they have a similar effect that temperature does, which acids especially can affect the three-dimensional shape of a protein. So if you get too strong of an acid, then you can actually have an effect on all the proteins that are exposed to that acid. So it's a similar disruption that temperature has. So it breaks the three-dimensional structure. And really, most life on Earth occurs in a pH range that's considered neutral. So pH is a funny thing. Scientists are funny people, right? They don't always explain things in an intuitive way. So here's another example. So pH, it's all screwed up. But the way it works is you have a neutral pH, and a neutral pH is where you have a balance. And let me just bring this up to really understand pH. What you have to understand is pH actually, it kind of comes from water, and water is an H2O molecule. We're familiar with water. Well, really what an acid is it's an H. It's got a charge to it, so it's an H+. And a base is an OH, and it's got a negative charge to it. And if you put these two together, H2O, you get water. So really, acids and bases are products of water, or in other words, they're the building blocks of water. So just to clarify, an H+ is the constitutive molecule of an acid, and an OH negative called a hydroxyl is the constitutive molecule of a base. So what I was saying earlier is as you increase H+, you increase the acidity. And so really acids are high concentrations of H+. Okay, so why I said all that is because when you have an equal amount of H+ and OH, you have pure water. And really, pure water is a perfect mix of those two, H2O. And if you have nothing but pure equal amounts of those two, you essentially have pure water. I'm sure if any chemists out there get a hold of this, they're going to be screaming at the top of their lungs at what an idiot and how poorly I'm explaining all this. But that's basically water and in relation to acids and bases. Now, acids then become a substance with a higher concentration of H+. So as you start to shift that balance and you get more and more H+, you essentially get a stronger and stronger acid. Now each whole number up or down, so if we're going in terms of acidity, so to clarify, we're going down on the pH scale, so we're going from 7 south down to 6, 5, 4, etc. So that would be something acidic, lower than 7. But each whole number actually represents a tenfold increase in the amount of acid, in this case, acid or base. So if we're going down, the difference between a 7 and a 6 is a tenfold increase of acid. So in other words, a pH of 6 is 10 times more acidic than a pH of 7. And that then it works in that a pH of 5 is 100 times more acidic than a pH of 7. So you multiply by 10 each time you go down. So a pH of 4 is 1,000 times more acidic than a pH of 7, etc. And the key there is that there's a big difference between a 7 and a 6 and a 7 and a 5, etc. So that's why the pH scale's all screwed up. None of that makes any sense, but that's ultimately how it works. And the same is true if you're going up. So a base is something that has a higher concentration of OH negative. Okay? And in a similar way, too much of that starts to disrupt proteins and causes a disruption in the shape. So generally speaking, as you go up too high, that has a negative effect, just like going down too low has a negative effect. And again, each whole number up represents a tenfold increase. So anything between 0 and really say 6.9, just below 7 really, will be considered acidic. Exactly at 7 you have neutral. Anything above 7 all the way to 14 is considered basic. Now one more thing to point out here. Most organisms only exist in a fairly narrow range between a pH of 6 and 8. So generally speaking, life can only exist in an environment with a pH close to neutral. So if we go too far south or north of neutral, it generally becomes toxic. Now, if you look at human blood, for example, the human body has a pH of about 7.4, and it's pretty specific. And if you go too far outside of that range, you can actually become toxic and your body doesn't function and you can actually die. So our bodies regulate that pH, but ultimately too much acid or too much base and our biological functions would no longer work anymore. Now, the reason you don't die if you drink tomato juice, which has a low pH, is because your body can balance that pH and buffer it. And if you want to know more about that, you can look up buffers. And a buffer is something that balances a pH. It makes it so that it doesn't become too acidic or too basic. But I'm not going to get into that here. So that's how pH works. Now, and just like in temperature, some enzymes have a preference for higher or lower pH. So most enzymes function in coordination with the 6 to 8 range. There are exceptions, and this is an interesting exception here, where we have an enzyme that functions in our intestinal system called trypsin. So this functions in the small intestine of our body, and the small intestine actually has a slightly higher pH. And so actually in our stomach, we have a really low pH. If you go back to that figure here, this is actually showing the gastric juice in our stomach, which has a pH of 2. So to clarify, each part of our body can have a different pH. This is showing urine, this is showing the stomach, this is showing human blood. So it's not necessarily one central value, but in the small intestine we have a slightly higher pH than we say we have in the stomach, in our stomach cavity. So an enzyme like trypsin functions much better at the closer to optimal pH. Showing it functioning optimally here, right around a pH of 8. Whereas a different enzyme like pepsin functions much better at a lower pH, closer to that of the stomach, where we have lots of gastric juice. So different enzymes function at different pHs, and generally speaking, our body is adapted to allow different enzymes to function in different sites. So trypsin is meant to function in the small intestine, pepsin is meant to function in the stomach, and therefore the pH kind of is conducive to each environment and allows each enzyme to function as it should. The stomach, by the way, is incredibly acidic. We're looking at a substance that can almost eat through metal, and we carry that around in our stomach all the time. So it's amazing how acidic that really is. Okay, now the next part here is to go back and connect enzymes in disease. And then we talked about this in chapter 13, so I don't want to reiterate the whole thing, but just want to make sure to connect that back. So when we talk about an enzyme, they all have a substrate, and the substrate is going to get reacted upon. Now, when it comes to pathogenic enzymes, these are generally going to be catabolic enzymes. And what happens with these is they break down natural tissue in the body. They break down tissue in a living organism. So in this case, what ends up happening is the substrate is host tissue. Our body becomes a source of substrate for these types of enzymes. And what a pathogen is essentially doing is breaking down your body in a way that causes destruction and problems with the host tissue. Sorry, my cat is bothering me. But generally speaking, these types of enzymes are referred to as virulence factors. Now, virulence factor is anything that causes damage or is considered contributes to the virulency of a microbe. But many of these are considered enzymes. So there's something that reacts with the host. So here's some examples from chapter 13. And go back to that section if you want more detail. But mucinase breaks down mucus. And if you remember, mucus is a protective layer. So mucinase is a pathogenic enzyme that allows a pathogen to break through that protective layer. So they can literally digest it away. So that's ultimately what makes an enzyme, or excuse me, what makes a pathogen is the fact that they produce certain enzymes that react negatively with you and I as their potential host or other organisms. And really what it comes down to is kind of an unintentional interaction in which the way that their metabolism works doesn't coincide with our metabolism very well. And as a result, they end up causing harm to our body, and we can't coexist with them. So that's really kind of how that works. So anyway, here's part 4, review part 4. So go ahead and pause it here. We got one more section. The last part's pretty short, so we'll be done here pretty quickly. Okay, and so to elaborate on the pathogenic enzymes and what makes a pathogen, generally speaking, we can look at most microbes as either a parasite or a saprobe. And we talked about the term saprobe back in chapter 5. This we use this in reference to fungi. We said many fungi are saprobes, and a saprobe is a decomposer. Generally that term describes them as a decomposer, but it also kind of implies that they're relatively harmless and not considered pathogenic. So generally a parasite is harmful, whereas the saprobe is harmless. There are definitely exceptions to that, but that's kind of the general rule of thumb there. So so one of the general differences between a harmful and a harmless microbe is the types of enzymes that they produce. So if they produce an enzyme that interacts with their host in a negative way, then it generally makes them a parasite. If they don't produce any enzymes like that, and their enzymes don't have any negative interaction, then they're not harmful, and we refer to them as a saprobe. But really, when you get down to it, that's really the difference. And I'm generalizing, and that's not the case in every scenario, but that's a pretty good way to think about it, and that kind of summarizes, in a kind of a simplistic way, the difference there. And so really, a lot of the parasitism is just metabolic things that don't coincide. Their metabolism conflicts on our metabolism, and so they end up causing harm. Now, parasites are not just a straightforward thing. So there are different shades of different degrees, rather, of parasitism. So going back to the difference between opportunistic and primary pathogens, the truth is they both produce harmful enzymes. The difference is that one can survive the immune system better than the other. So opportunistic don't usually survive the immune system, whereas primary pathogens can. They're more capable of that anyway. So they can ultimately both cause harm, is the point there. Sorry, I lost my train of thought. But both can cause harm, but the difference is whether they can survive your immune system long enough for them to do any harm. Okay, hopefully you remember all that. I know we talked about that a lot, so I don't want to keep beating that over the head here. So anyway, that's one of the major differences. So last little thing here, microbes digest things in a really unique way, and there's some consequences for that. So this is out of chapter 7, and what more advanced organisms do is they phagocytize. Remember phagocytosis, where the cell engulfs a molecule and eats it. Many larger eukaryotic organisms do just that. They engulf and they swallow things. Smaller microbes like bacteria, many fungi, and things like that, they don't do that same process, and they don't have the same ability. And so what they end up doing is they actually digest the food outside of their cells and then absorb it once it's broken down. So the organic food substance is too big to simply swallow or absorb somehow. They just don't have that ability. So what they do is they release all their digestive enzymes outside of the cell, and they break it down externally, and then they absorb it in smaller chunks, shown here. So it's a way to get around being unable to absorb larger particles. And this is how organisms actually began life. These are the simplest organisms. So ultimately that ability to engulf food came later. So this is really one of the more primitive ways that it takes place. So, that's in itself a concept. Microbes digest food external to their body. Exceptions would be things like protozoa. Protozoa would phagocytize in most cases. So generally speaking, things like bacteria, most fungi would feed in a way like this. So ultimately break down and then transport back into the cell. So let me just go back here real quick. When it comes to pathogens, what they're doing is they're digesting your own tissue. So rather than being organic debris, it's human flesh. But the microbe releases an enzyme, it breaks down the flesh, and then it can absorb the nutrients from your own tissue. Now, they break down tissue for different reasons. Sometimes it's to get nutrients, sometimes it's to spread through the body. We talked about this in chapter 13. But either way, they're releasing enzymes and breaking down human tissue, and that's what causes disease. Now, in the case of a saprobe, one of the reasons why they're considered a decomposer is that rather than breaking down living tissue, they're breaking down organic molecules in the soil. So, for example, when an organism releases waste, feces and urine and hair and dead skin cells and snot and everything that comes out of a human or an animal's body, all that stuff goes somewhere. And ultimately what happens is it has to be broken down. And this is where tiny little soil bacteria and fungi that we never even know are there, they take those relatively large chunks of stuff, fecal matter for example, and they break it down into tiny, tiny, tiny little pieces. And they do that by releasing their enzymes onto it. And what that does is it starts to break it down. Now, they then absorb a lot of that, but they don't ever absorb all of it. And so I like to think of these as like a baby with a birthday cake. And the baby is going to eat some of that cake, but most of it's just going to go all over the place. So they're super messy. They're not at all efficient. They don't absorb most of what they break down. And so as a result, they're just breaking things down, and a lot of that these smaller nutrients here end up just kind of getting tossed into the environment, which is a good thing, because what that does is it adds nutrients to the soil. You can think of these as nutrients. So whatever the bacteria or microbes don't absorb basically just becomes a source of nutrients in the soil. So they're taking all this waste and they're generating nutrients from it. So they run off of it, but then ultimately what ends up happening is other organisms absorb it too. So things like plants on land and algae in the water become the beneficiaries of this. And so these nutrients are things that they use to drive their own cell processes, things like nitrogen and phosphorus. And really you can think of this as like fertilizer. Really, fertilizer is just that. It's the nutrients that these organisms create. So if you're buying organic fertilizer, you're buying the byproduct of all this breakdown. That's why people use cow manure and stuff like that. It's got, it's been partially digested by all these microbes, and it creates all these great nutrients. So saprobes are in decomposers, which again are pretty much the same thing. They are important because they generate most of these nutrients, and without them we wouldn't have successful plants, we wouldn't have algae, and we wouldn't have nutrients available to us in the soil. So if they're a pathogen, they're bad because they produce enzymes that break down living tissue. If they're a saprobe, they're good because they create nutrients in the soil through decomposition. And ultimately that aids in the process of plant life, which is good for us as well. So that is, in a nutshell, how enzymes relate to this, what's happening in a cell, and then how that relates to pathogens, and then how that relates to the environment. Okay, so hopefully all that makes sense. As always, send me an email. Let me know if you have any questions. Hope you have a great week, and I'll see you next time.